Good morning, everybody. My name's Josh Dahl. I'm the CEO of Singular Genomics, and I'm excited today to kick off the first annual Spatial Summit, in which we've brought together over the course of the next 2 days leaders in AI and precision medicine to help you understand how they're using the G4X, our in situ spatial sequencing platform, in order to better understand disease biology and biomarkers. We have some incredible speakers lined up over the course of the next 2 days, including Giannis, Taehan, Shani, and Evan. They come from prestigious academic institutions that study AI and precision medicine, and they're at the intersection of spatial biology and where the field is headed. And we're extremely excited for them to help you better understand how they're using the G4X to start performing spatial multiomics at scale. It's no secret that spatial technologies are gaining traction and people view them as having great importance, whether it's transcriptomics, proteomics, or now the intersection of the two with multiomics. um, People are doing some pretty amazing things, and that's really highlighted by the rapid explosion of publications in the space, whether it's from Singular or some of our friendly competitors. There's a lot that's being done in the space, and I think everyone's starting to see that as the cost comes down and the throughput of these instruments increases, there'll be an ever increasing need for clinical applications where now 89% of spatial users are believing that spatial technologies will find their way into the clinic. And we're really excited to help drive that transformation. So, what's the difference? Traditional pathology, whether it's anatomical or molecular, you're always sacrificing something. You either have low resolution while retaining spatial context in you know, the applications of IHC or fluorescent H&E or just traditional H&E. In addition, on the molecular side, you give really high resolution. NGS, I think, is a great example of something that's driven hard into the clinic, but you lose all of that spatial context. And I think the beauty of what spatial in situ sequencing is going to offer is that you can retain that high resolution, but also retain spatial context, and to give you insights into all of these different data layers and be able to interrelate them to one another. And that's incredibly powerful. And so, really the problem is today is that whether it's AI model development or translational applications, you really need to run thousands of samples in order to address heterogeneity. And without being able to do that, it's really unachievable to make translational insights. And, it can be done. It can take years and it can take millions and millions of dollars. But I think with newer technologies, including the one we'll share today with the G4X, I think we can start to transition this into something that's more achievable for most in the field. And so, really, what we're trying to do at Singular is deliver spatial pathology at scale. And with that, I'll turn it over to Darius, who will go over the G4X platform before turning it over to our collaborators. Thank you.
All right, thank you, Josh, and thank you everyone for joining us today in our inaugural Spatial Summit. So if you've been following us over the years, you might be aware that the team at Singular has developed a powerful sequencing engine completely in-house. This has been an 8 to 9 year effort, starting with the underlying chemistry, which is a 4-color SBS chemistry. That's paired with rapid fluidics and high-speed imaging to process over 2 billion pixels per second. And really, over that time period, the team has advanced that capability from standard next-generation sequencing into in situ sequencing directly in cells, first in a 96-well glass plate, and then eventually moving that into tissue and eventually into FFPE, or archival material. And so today, what we have is the G4X. So this is a high-throughput in situ multiomics technology, and there are a few things that make this special. For one, is the throughput. This is about 10 times the imaging area per run, with over 40 centimeters squared and up to 128 samples that the user can process per run. The nice thing about this throughput, besides the speed to execute large studies, is the cost profile, and this allows us to move the field from thousands and thousands of dollars per sample down to hundreds. And the system has integrated multiomic capability, able to process transcriptomics, proteomics, and a digital H&E in every sample. And this all comes without sacrifices in performance. We're able to get precise single-cell results from FFPE material, even the most challenging samples, all at subcellular resolution. And last, we have a novel capability called direct-seq. And this is really exciting because they can do things like in situ CDR3 sequencing or sequencing of other variable regions, think cancer hotspot mutations or CRISPR edits, that you can weave in with the other readout modalities. And this is still in R&D, but very, very exciting. So a little bit more on those readout modalities. We've got, starting at the bottom, on every FFPE sample, we do a H&E. This is eosin's already fluorescent. We use a fluorescent version of hematoxylin, but this it looks like a standard H&E, allows you to visualize the tissue morphology and connect into standard histology and maybe digital pathology data sets. We also in the same run do a 17-plex proteomics. So this is amplification-based approach. These are oligoconjugated antibodies with rolling circle amplification. And then we do a single base imaging. We do multiple rounds of that. This allows us to get to that 17-plex. It's all at very high sensitivity. In that same run, we do 500-plex transcriptomics. So this is a barcode-free approach, which carries inherent benefits that Mike will touch on later. But this allows us to look at 500 gene transcripts with very high sensitivity, specificity, and reproducibility. And then direct sequencing is that last modality that we're working on in R&D. This allows you to look at immune repertoire sequences, cancer mutations, gene editing, all within spatial context of the other readout modalities. Very powerful, as you can imagine, looking at clonal expansion of B or T cells or certain clonal expansion of cancer mutations in context of the rest of the tumor microenvironment and immune response to that. There's very powerful potential translational applications of that. So that's still in R&D phase, but the technology that we're largely talking about today is capable of these bottom three modalities: the 500-plex transcriptomics, 17-plex proteomics, and fluorescent H&E. So a little bit about the workflow. We have, first preparing for a run. Users can quickly and cost-effectively customize panels, either building from our base set of off-the-shelf panels or creating their new ones from scratch. We have a patented high-throughput tissue transfer that allows users to go from a section off of an FFP block onto an arrayed set of samples on the slide through a process that we call tissue stamp, and you're seeing the outputs of that here on the left. And that actually, there's a session later today that details that more specifically. And then there's overall, it's about a 3-day workflow that's been streamlined. So the run itself, you can process between 4 and 40 centimeters squared per run across 4 flow cells and up to 16 fluidically independent lanes that you can run different panels in. The run itself is a 29-hour image acquisition, and then analysis right now is a couple days after that we're working on accelerating. And then this is all done on a field mature underlying platform. We've gotten years of user feedback on the sequencing side, so that we've been able to incorporate. And this is really nice. This means that we're not having to launch this on a new system and all the kinks associated with that. And then last, the analysis. We do all single cell analysis, so onboard cell segmentation, so results are produced at the single cell level and all of our comprehensive QC metrics. We have a web-based multimodal viewer. It allows you to visualize the H&E, the proteins, the transcript layer, and the cell segmentation mask all together with cluster identification, nice way to navigate the tissue and get some nice images. And then we have standard spatial file outputs, really for your kind of preferred downstream tertiary analysis pipelines, whether that's open source or commercially provided. Most importantly, the G4X gives the field unprecedented scale without sacrificing data quality. And I'll provide two quick examples of what scale can do for researchers. The first is 3D analysis of tissues. So here we performed a 50 micron reconstruction from 10 serial sections of renal cell carcinoma in a single flow cell. And this reconstruction resulted in 6.2 million cells with both transcript and protein information at subcellular resolution, which allowed us to do single cell phenotyping and cell neighborhood analysis not only in the X and Y dimension, but also in the Z dimension, enabling us to precisely see how the tertiary lymphoid structure was interacting with the tumor across all dimensions. And this data set is available on our website. You can download the poster in which we go through this in more detail. But we believe this is impactful because biology is inherently 3D, and we believe that the G4X is uniquely capable of delivering high-quality data sets for tissue atlasing programs, providing researchers a deeper understanding of the tissue microenvironment. And second, and equally important, is large cohort analysis. So here you're seeing that the G4X is capable of processing 128 samples per run across 16 fluidically independent lanes and powering large translational studies. So these large-scale studies are absolutely fundamental for training high-fidelity AI models and developing translational insights from those across heterogeneous sample populations and bringing the whole field closer to impact and better diagnostics in precision medicine. The scale on the platform is also enabling us to finally make spatial analysis affordable, which we believe will enable the field to move from small-scale studies into studies that can start impacting precision medicine in a more meaningful way. So for transcriptomics and fluorescent H&E, our smaller 4.5 by 4.5 millimeter samples are $240 per sample, and our larger 10 by 10 millimeter samples, which you can fit 10 per flow cell, 5 per lane, are $768. As this spatial information is all at single cell readout, this equates to really an unprecedented 0.08 cents per cell. And if you want to run multiomics, so adding our standard 15-plex protein markers on there, this brings up the price a little bit, but for $310, you can get information on our 4.5 by 4.5 millimeter sample and under $1,000 for our bigger 10 by 10 millimeter samples. But really beyond reagent pricing, if you consider CapEx and lab operations and other expenses, we estimate that the G4X can result in about a 5-fold reduction in cost for a spatial at scale. So we've had a productive year so far at Singular. We've completed a successful early access program with 5 sites, including Beth Israel Deaconess Medical Center, Vanderbilt University Medical Center, Stanford Medicine, Cedars-Sinai Medical Center, and Albert Einstein School of Medicine, some of who you'll hear from this Spatial Summit. Internally, we've completed 18 technology access cohorts processing over 20 FFP sample types as shown on the right between mouse and human, including notoriously difficult samples like bone marrow, where we got our first publication in Science Immunology with Dr. Catherine Wu out of the Dana-Farber Cancer Center in AML. And in total, we've processed over 10,000 FFP samples this year on the platform and over 2 billion single cells. And by our estimates, this is the largest such data set on the planet. So if you're interested in helping us shape the future of spatial pathology, there are a few ways to get involved. As we move beyond our early access sites, we're now starting to stand up the technology at our next 10 to 15 sites. These are primarily top academic medical centers who share our vision for the technology and the application. These sites will help lead the rest of the field for the technology. And as we roll out more products on our roadmap like DirectSeq, there'll be early users of that as well. If your institution is earlier in that journey, we're running technology access services where we can generate G4X data on your samples to demonstrate the performance of the platform to you. And then lastly, if you're just exploring, feel free to download one of our 8 publicly available data sets. to analyze yourself or play around with in our viewer. So in between some of the keynote talks today and tomorrow, you'll hear more detailed information about the technology from members of our team, including some of the latest performance data. And we're always happy to chat if you want to reach out and talk to an expert on Singular's team after the summit. So with that, I'd like to hand it over to Dr. Yanis Vlachos. He is the Associate Professor of Pathology at Harvard Medical Center and Director of the Spatial Technologies Unit at Beth Israel Deaconess Medical Center. Yanis was the first G4X early access site and has been a great collaborator. He'll be telling you a bit about his experience with the platform and his talk, High Throughput Spatial Sequencing, Redefining Limits and Workflows in Basic Research, Translational Medicine, and Core Facility Services. Thank you.